we have thus far covered the structure of the membrane, the form of the membrane, and thus the function of the membrane. We talked a little bit about how the membrane formed, and we've talked about passive transport that doesn't use any energy to get molecules from one side of the membrane to the other, following a concentration gradient, moving from a high concentration solute to a low concentration solute. In this mini lecture, we're going to focus on going the other direction across the membrane, where we go from an area of low concentration solute to high concentration solute. And that's hard to do, so it's going to require some energy. Thus, we're talking about active transport. Now, most solutes, as we know, cannot get across the membrane all by themselves. They need help. What will help them across are transport proteins. These alter the permeability of the plasma membrane. They make it so that more individual molecules can get through. One type are the channel proteins. These are not very specific. They sort of create big open holes in the membrane. If the molecule is large enough to fit through the hole, it gets in. These molecules moving through channel membranes follow their concentration gradient. On the outside here, that extracellular fluid, you'll notice it's got a higher concentration of solute. That high concentration of solute wants to diffuse to the inside, where there's a low concentration of solute. If you want to be more specific, you're going to use a carrier protein. Carrier proteins are going to grab a molecule from one side of the barrier and then selectively pull that one through and drop it off on the other side. There are three kinds of carrier proteins: a uniporter, symporter, and an antiporter. For a uniporter, you are simply moving things from one side of a cell to the other. That's it. Whole thing. I'll show you again. I'm the uniporter from here to there. Questions? Uni means one. I can move one at a time. A symporter. With a symporter, you have two proteins or two molecules moving in the same direction. So here's the membrane. I'm the symporter. I bind to one first. That opens up a binding site on another place. It binds, which allows me to drop both off on the other side. So again, I can bind once. That opens up. I can bind another, and then drop both off. One binds first, and then the other binds. You have to have two binding at the same time, two bound at the same time. That maintains ratios. So if you're making salt, now your cell's not going to make much salt, you would take in a sodium ion and a chlorine ion so that you'd always have the same ratio of sodium to chlorine.
What does it
Bind, hold on to.
Like, what is it binding?
The transporter is going to bind to the whatever it's on the outside that wants to bring in. If I needed to bring this into the cell, I have to bind to this. So it's the
She's asking, what is that supposed to be?
that's nutrients, ions, anything the cell needs. I'm sorry, thank you. Appreciate you clearing it up.
Part of a cell.
Part of cell food. Yes?
Could you do the first one again?
The uniporter? Sure. With the uniporter, it binds, carries it across. Thanks for letting me use your stuff. A symporter, two things on the same side have to bind together before they're allowed to be brought across. With a symporter, two items from outside the cell have to bind, and they're brought through to the other side.
So you're taking things from outside the cell.
Bringing them in, right. And then an antiporter is where you have two molecules moving in different directions. So for this to work, you bind on one side of the cell. That opens up a binding spot on the inside, and then they're able to switch.
So they're moving against each other. Anti. Sure. So first, you have a molecule bind on one side. That opens up a binding site on the other side. So now I have one on the inside of the cell, the other's on the outside of the cell. And then after they've both bound, they switch places. Why not just have like two one-way ones? Is that the question? Why is there three different ways of getting it through? So a uniporter is going to constantly, it doesn't matter the amount that's inside, it will just keep bringing it in. With a symporter, you're going to bring in equal ratios of two things. And why that's important is, so if you're making salt, hypothetically, salt's an easy one, NaCl, right? If you're making salt, you don't have 1 sodium and 15 chlorine. Does that make sense? You don't need 15 chlorine, so you only need them in a 1-to-1 ratio. So you bring them in together in that exact ratio, and you never have too much. And then with an antiporter, you can use it in order to, one, get rid of, every time you get rid of waste, you're pulling in something new.
So these are the 3 types of carrier proteins, and what are they carrying in again?
They're carrying in ions, they're carrying in nutrients, they're carrying in vitamins, they're carrying really just about any molecule. Yeah, carbohydrates, nucleic, well, not really nucleic acids. They'd bring in sugars, things like that. No problem. Uniporters, symporters, antiporters. Now, as long as you're following your concentration gradient, that does not require energy. It happens naturally. The problem is when you go against your concentration gradient. That's when you require energy. So we get, thus we beat on, boats against the current, borne back ceaselessly into the past. Thank you. That is the Great Gatsby. In case you guys didn't know, that's the Great Gatsby. My first semester teaching, I made the mistake of saying, hey, if you know what that quote's from, I would give you an A in the course. And then I realized, oh, that'd be a horrible thing, because then I just have to give Amber an A. So I stopped doing that. But you do have my sincere appreciation. The Great Gatsby was a wonderful book, and great life lessons there. Don't look back into the past. Don't reach out for the green light at the end of the dock. The big thing is, boats against the current. If you are in a canoe and you're on a river, it takes barely any energy to move forward with the current. It takes a lot of energy to move against the current. If you've ever been in a crowded city, and I don't know that Richmond's that terribly crowded of a city, if you've ever been in like New York trying to walk against the crowd, it requires energy. Going with the crowd doesn't.
So I'm confused, like how does that relate to what we were just talking about?
How does active transport or the...
So
So we were just talking about those transport proteins, right?
So are you saying which ones of these is like easiest to do?
If that transport protein is moving with a concentration gradient, it does not require energy.
Okay.
If, though, you're pumping against it, if you're using a uniporter, for instance, to push a molecule from an area of low concentration to high concentration, that's going to require energy. They naturally follow that concentration gradient, but you're making them go against it. So that needs energy. That needs ATP. Any of them will if they're going against the concentration gradient. Anytime you move from an area, you're trying to push solutes from an area of low concentration to high concentration, you need energy to do it.
So that is, we're looking at diffusion there, not osmosis.
Correct. If we were looking at osmosis, it would be the movement of water, and these are not water. These are not water molecules. Yeah.
Osmosis used when we're talking about the
We're talking about the membrane, and we're talking about going directly across it using water.
The water is just one aspect of it.
Yes.
We also use different solutes and things.
Right. So now we're talking about a different way of. So we talked about getting water across using osmosis.
But is osmosis and diffusion happening at the same time?
at the exact same time. You have a lot of processes happening at the same time. Sometimes we can use a pre-existing gradient to power a pump. We can use the power of a gradient to provide energy for our pumps. This is called secondary active transport. This is sort of like if you're a jerk, hypothetically. You guys have used revolving doors before? Yeah. How does a revolving door work? You don't use it. So you go counterclockwise. You go to this revolving door and you push on it. That pushes the door around and you can go through, right? But you don't have to actually push on it. How can you get through it without pushing on it, without expending any energy? You stand and, just like Isaac said, you stand and you wait, because what's going to happen eventually? That's right. Somebody's going to come from the other direction. And if they come the other direction, they're going to push on that, and that lets you get through without you expending any energy. So it's passive on your part. Same thing happens with molecules. You can move from an area of high concentration. You have some molecules pushing through, and as they push through, like with, say, an antiporter, they're pushing in one direction, so another molecule can get pulled through in a different direction without the use of energy, because you're using the energy of the other gradient. Can it ever just go too fast so nothing gets in? So nothing gets in? I don't think so. I think these things move very, very efficiently. Now, if you combine this with ions, so you have more ions on one side of a membrane than on the other, you've got an electrochemical gradient. You're using that spinning action in order to provide energy to power reactions. Literally, you would have water wheels here that are spinning and creating energy. So you can form electrochemical gradients. Those gradients can then power the creation of what are termed energy intermediary molecules. This becomes the basis of aerobic cellular respiration, which we'll get into in a few weeks. So it's not terribly important for now. It will be terribly important later. Some molecules are just too big to move through channels, too big to fail. Nucleic acids are huge. Certain proteins are gigantic. In order to move those through the membrane, you have to use a process called either exocytosis or endocytosis. Exo means out, cyto is cell, and osis means action, so outside cell action. What happens is we've got, for exocytosis, we're packing things up in the Golgi apparatus for secretion, individual molecules. Those are being sent in a vesicle over to the membrane, where they're released to the outside. Endocytosis works in the exact opposite way. A particle binds or gets close to the membrane distorts, actin filaments slide along, and you end up engulfing it in a vacuole. So you take in a vacuole. So what you can do, you could be packed up in the Golgi apparatus. You go out, you hit the membrane, it fuses, and then opens up and pushes it out. The other way it goes, you could have it come in and bind, a vesicle forms around it and pulls it in. So that's exocytosis and endocytosis.
We started off this mini lecture talking about different classes of proteins that transport solutes across a membrane. We talked about transmembrane proteins. Transmembrane ones cross the whole membrane. Usually they have this sort of open center, or they allow for molecules to come in and then move to the other side. They could come in different varieties. You might have a symporter, a uniporter or an antiporter. A uniporter moves one molecule across at a time. A symporter moves two molecules together through, or an antiporter moves two molecules in opposite directions through the membrane. We talked about active transport requiring energy, needing some sort of reaction to occur to give it energy and push solutes against their gradient from an area of low concentration to high concentration. That's super hard to do. There are varying ways of doing that. We might use secondary active transport, where we're using various pumps to move two molecules at a time across in sort of an antiporter mechanism. When we can't get molecules across through a transport protein because they are too big, and there are a lot of molecules that are just far too large, they could get across the membrane using special processes like endocytosis and exocytosis. In endocytosis, the membrane deforms and sort of creates a little pocket in the membrane that a particle will go into. That pocket will close off, and then you'll have a vesicle inside the cell. And with exocytosis, you're moving in the opposite direction. You take a vesicle in the cell, it fuses with the membrane, opens up, and releases its contents out of the cell. These content review questions are there to really focus your studies, make sure you understand them in order to, keep moving ahead in the course, because every concept builds on each other. The next mini lecture is going to be about how cells communicate. So the membrane, the exterior part of the cell, is the primary mechanism by which different cells are able to communicate with each other. It's how they engage with the outside world. And we're going to really delve in depth on how a cell can send a signal, receive a signal, transduce that signal, and then have a reaction. So stay tuned.